So hi everyone, my name is Phidias Timandis, and I'm a neuropathologist at the University Health Network and the University of Toronto. And we're going to be continuing our histopathology series, talking about the most important stain of all, the Hematoxylin Eosin stain. We're going to be talking about some of the principles and some of the future directions for this historic stain. So again, just as a disclaimer, this is not for diagnostic use. This is purely for educational improvement. And so use this information with caution, and of course, supplement it with information in your own training program and jurisdiction. So we're going to be talking about the Hematoxylin Eosin stain, which is often abbreviated as H&E stain. And we're going to be going over some definitions. We're going to be looking at some of the competing or complementary technologies for this stain, which include histochemistry, immunohistochemistry, which is the subject of another NeuroPsyQ video, and just talking about some of the key differences with molecular, which is kind of the kind of modern tool that we have in pathology that's often being compared to histomorphology. We're going to go look at specifically the theory and principles of the H&E stain, including tissue processing, preparation, sectioning, and staining. And we're going to do this for H&E, and then we're just going to quickly build on some of the variants, such as the H&E LFB stain, and just quickly touch on kind of histochemistry stains as well. We're going to go over briefly go over the limitations, which is primarily the subjectivity of the H&E stain, and how some modern techniques like artificial intelligence and computational histopathology are really revitalizing H&E stain as a very powerful tool in the modern era of precision medicine. So we'll kind of quickly go over some of these concepts, which are kind of are discussed more in detail in other episodes found on this channel. And here we have some images, so we might as well discuss them. This is both normal and infarcted brain tissue. So this is what happens in a stroke. So on top, we see that the H&E stain is identifying these different types of cells. We have this pink area in the back. This is largely processes of other neurons and glial cells. We have these kind of round cells, which in the brain are usually oligodendrocytes, especially when they're positioned very closely to the neuron like this. And then here in the infarcted brain tissue, we see that the neuron has completely lost its kind of resolution of its nucleus. It's become pink. We can't make out many of its internal structures. And then there's a number of infiltrating neutrophils with these multiple lobes that indicate that this tissue is dying. The neurons are dying, and the neutrophils and other inflammatory cells are coming in to clean up the mess. So as you can see, very powerful tool. Even at this high magnification, we can quickly understand what's going on in this particular section. So what did we just do there? What's the definition of H&E or histomorphology? It's really the microscopic analysis of cellular patterns and architecture. So how, what type of cells are in tissues, as we saw, and what architecture are they forming? We can learn a lot by the specific structures that cells form by interacting with themselves and other cells. And this is really the basis of histomorphology. As I mentioned, there's some subjectivity, as you can imagine, of what we just did. Some pathologists may interpret what I saw as differently. And of course, the same pathologist can also interpret the same slide differently, depending on their specific level of concentration, workload, and kind of other cases that they've seen in their recent memory. So, this is one of the kind of critiques of histomorphology, where it should be kind of improved by other techniques. And one of those has been molecular, but it's important to understand that these are categorically different. And in the modern era, these more complement than compete with each other. For now, we'll just say molecular is more objective, it's more precise, but the kind of advantages that histomorphology brings is speed. You can prepare an H&E section within minutes to days, minutes being in the operating room during an OR, and days, which is really our routine kind of turnaround time for preparing H&E sections. And you compare that to days and weeks and sometimes months for very subspecialized molecular tests, you can see that, you know, if you're a patient, the last thing you want to do is wait weeks to months to have a kind of fairly good idea of what's going on. The cost is also a problem. An H&E stain can be done for a couple of dollars if you have the right reagents and setup, whereas molecular tools usually require, even with investment into the infrastructure, they still require hundreds to thousands of dollars per test to complete. The histomorphology is very versatile. It can work on, as we saw, infarctions, so you can detect chemical changes in oxygen levels. It works for infections. It works for tumors. It works for any number of different disease types, neurodegenerative conditions, whereas many molecular tests that we run today are highly specific for a specific use case. So you must know what exactly you're looking for before you use those molecular tests. So the versatility of the H&E stain is quite remarkable, and is probably the most dynamic stain, in my opinion, in all of pathology. And again, these are complementary, right? Certain things you can pick up with genetic mutations, but other things like hypoxia, inflammation, may not appear on kind of genetic sequencing. So these are in fact complementary. And on the reverse side, there's some genetic events that are really important, like MGMT, methylation, promoter methylation in glioblastomas. This is probably something we'll never be able to resolve with histomorphology. And so the two serve a different purpose in the diagnostic work of cases, and should always be thought of working harmoniously together rather than competing. One of the differences is also that histomorphology is subjective. This may be seen as a kind of a con, but for someone investing a lot of time in their training, being able to look for things in a kind of unsupervised way based on your experience is actually quite powerful. It's harder to be displaced from the workflow through automation, but also allows you to discover things that may not be built into a kind of a diagnostic algorithm of specific genetic changes. And the other amazing thing about the H&E stain is quite robust to variations, even though there's different protocols and ways to do the stain. The overall result is very similar and can be quickly kind of climatized if someone's doing the stain somewhat differently. So lots of advantages of why this stain has kind of stood the test of time. Okay, so what's the bread and butter? Hematoxylin Eosin. Really, what we're looking for is looking at the global set of architectural patterns under the slide. Kind of look for specific groups of diseases. Is this infectious? Is this degenerative? Is this neoplastic? And then use the set architectures to give us a better sense of subclassifying what we're seeing under the microscope. Is this an ependymoma versus a glioma? And of course, those have different prognostic and therapeutic implications. So lots of information that can be gathered from the H&E stain. Histochemistry sounds very similar, but the difference is here, we're not so much interested in the actual pattern. We're more interested in the presence of a specific tissue component, such as collagen or reticulin. And so it's kind of a yes or no answer as compared to a kind of more artistic impression of what we see under the H&E stain. One example of a histochemistry stain that we do often is reticulin. Many tumors are reticulin rich, whereas others are reticulin poor. And so the presence or absence of reticulin can help differentiate tumors that may be on the differential diagnosis, but have different chemical properties. Schwannomas tend to have a lot of reticulin. Meningiomas tend to form these syncytial bonds between the cells that are depleted of reticulin. So there, we're not so much interested in the structure, but rather the presence or absence of a specific cellular component. Immunohistochemistry is kind of the modernized version of histochemistry. This allows you to use antibodies to look for specific proteins, their presence, and their localization within the cell. We have a very nice NeuroPsyQ lecture on this, so please refer to that for more details. But it suffice to say that this is a very powerful technique where you can look for the presence of individual proteins. Another technique that was deemed to have going to replace histology, given the kind of sensitivity and specificity of it. But again, it's become an essential complementary technique, but one that by no means replaced the H&E stain. It's not going to be focused in this lecture, but just so you know, there's also electron microscopy where you can look at subcellular structures and see their morphology and any abnormalities. This is kind of commonly done for muscle still. Used to be done for some tumors, but now immunohistochemistry has largely solved the tumor problem, but it's still a very valuable tool in muscle disease and diagnoses. So let's look at what the kind of pioneer of the H&E stain or cellular pathology had to say about it. And really, this is 150 years ago. So imagine the kind of insight that these pioneers had in this specific field. The body is a cell state in which every cell is a citizen. Diseases are merely a conflict of citizens of the state brought about by external forces. And this may seem kind of a fairly elementary statement to make, but Rudolf Virchow lived in an era where it was felt that the only way to make a diagnosis was to examine a patient. And this idea that you can take tissue, remove it from a patient, make a slide and look under the microscope and find a diagnosis was revolutionary at the time. Now, of course, we take it for granted, but in that time, this was a kind of a profound idea that obviously has turned out to be the backbone of modern medicine. And so here we see an example of this. This is a tumor. This is a glioblastoma. So even in this tumor, we see disorganized glial cells. So we know that there is a tumor, but the amount of layers that you can make out is actually exceptional. So as we saw in normal brain tissue in one of the earlier slides here, it's much more cellular. They're much more disorganized. So we know that the citizens have been invaded by a tumor. But when you look further, you see that these tumor cells are actually forming these kind of palisades around this pink area. This pink area that has no nuclei because nuclei stain purple is necrosis. And really we're seeing the cells kind of retreat from these hypoxic areas that are usually caused by thrombosis of nearby vessels. So what you saw that right there was the kind of level of detail that we can extract from these very cost-effective slides, really showing just how powerful histomorphology is at not only finding abnormalities, but also classifying them into disease types, and even making very accurate predictions about the oxygen status of this particular region to the individual cell level. So this is truly the first single cell technology that has been invented and has just so much power due to its ability to make inferences from the overall cytoarchitectural pattern within disease. Another important comment, I think, that Rudolf Virchow made was that cellular pathology is not merely at an end when there are no alterations in the cells. So can you have disease without having alterations in the cells? Chemistry brings clarification of living processes nearer than does anatomy. Each anatomical change must be preceded by a chemical one. And I think this is tremendous insight that someone 150 years ago who invented the stain could already concede that there are some genetic changes perhaps that could be occurring that may not be visible under the microscope and may benefit from complementary molecular analysis. So even the pioneer of the H&E stain left the door open for cooperation with other laboratory techniques. Another kind of the second part of this statement, which I think is important as we talk about artificial intelligence and computational pathology, is that he mentions that each anatomical change must be preceded by a chemical one. So here, here's an example of a glioma with very ugly nuclei. Some are binucleated, some are extremely atypical. And that's very abnormal. And when we see this, it usually indicates that you have a P53 mutation or a mismatch repair mutation. And so a mutation that we cannot see in kind of DNA repair, genome guardian genes will lead to an upregulation of kind of this defective protein, which we can make out by immunohistochemistry. But we can also see it by morphology. And this has been leveraged by many computational pathology papers to try to predict specific mutations in tumors based on these kind of dogmas where chemical changes are usually, if they're important enough, will have manifest as some sort of phenotypic change at the microscopy level. So very important principle if you are in computational pathology to understand that this can often occur, but doesn't need to always occur. So there are some mutations that we will never be able to probably resolve using morphology alone, but other ones we can leverage to make quick predictions about specific mutations and guide our immunohistochemical and molecular workup, because some tumors with mismatch repair mutations may need different types of molecular tools than ones without them. So it's important to understand this principle. Okay, so let's move on to sample preparation. A sample is removed and needs to be fixed. It needs to be fixed for a number of reasons. The tissue will undergo a process called autolysis. If the tissue is not fixed, it'll degrade naturally. And so we need to have a way to glue the cells and the subcellular structures together so we can examine them in a way that we're not under time pressure of the tissue degrading. And to do that, the process is called fixation. Fixation serves two purposes. It helps fix the tissue to preserve it, but it also helps as an important component to making the tissue more firm, which is needed for sectioning into thick sections that allow light to pass through so we can see it under a light microscope. So under a light microscope, we fix it with formalin, 10% neutral buffered formalin, 4% from formaldehyde. The key here is the percent is low. So what's the other percent is? It's mostly water, which is an important, it's important to note that in the other kind of preparation steps, that this is mostly an aqueous solution for this specific step. Electron microscopy, again, not something we're going to go into a lot of detail here, but the similarities here is that fixation uses another kind of aldehyde, formaldehyde, glutaraldehyde. So it's important to understand the mechanism of action of aldehyde fixatives, which we'll talk about in the next slide. But really, it's we're forming cross-links between proteins through the formation of these methylene bridges, which help keep the tissue and proteins where they were during their viable life. So by cross-linking it, we're effectively just freezing everything in time and space and allowing us to analyze them in a kind of longitudinal manner. The cross-links vary depending on the aldehyde used. So in formaldehyde, you have these CH2 bonds, whereas in glutaraldehyde, you have C5, H10, so longer kind of carbon backbones that are connecting proteins to proteins. It may be self-evident, but once you do this, you've basically destroyed the enzyme and protein activity of the cellular constituents. It's not a problem for most tumor analyses, but in muscle biopsies, this is important to understand because many of these stains rely on active enzyme activity. And so when you fix them, you may not be able to look at enzyme activity directly. The other important thing, mostly a historic problem, was that fixation does compromise RNA and DNA and proteins. So if you're looking to use these for research purposes or even diagnostic protocols, this used to be a major downside. This has been largely resolved with improved protocols that are FFP or formalin compatible. So cross-linking, again, this is not meant to make you a chemist or an organic chemist, but more just to understand the principles, just because a lot of these things happen behind the scenes in a kind of a modern, large pathology department. So we may not be thinking about the kind of fundamental biology and chemistry going on, but if you don't understand them, it'll be very difficult to troubleshoot with your technicians when something goes wrong. So it's important to understand the fundamental principles of which these fields are based on. So here we have a protein. It has this nucleophile, which is often a lysine amino acid group of a protein, and then it's going to react with formaldehyde, so CH2O, and the basic component of the protein is going to is going to cross-link to form this methylol group, and then this is going to lose water and form what's called a skiff base. This is obviously an unstable structure if you've taken organic chemistry to have these double bonds, and so at its earliest convenience, it will find another protein or DNA structure that has a nucleophile in the vicinity and try to form a single bond, allowing this cross-linking between a protein and another protein or DNA molecule. This can occur with different proteins. Protein DNA can occur with different regions of the same protein. So really, you have to conceptualize this as happening multiple thousands, millions of times within the tissue, allowing it to really firm and just keep everything exactly where it is. So once you fix the tissue, now it's time to start sampling. So there's two different things you may see in a report. In a pathology report, it may be denoted as being sampled in total, which means that the whole specimen has been put into pathology tissue cassettes, as we'll see. These are kind of 2 by 2 by 0.2, 2 centimeters by 2 centimeters by 0.2, 0.3 centimeters in height cassettes. So if the sample's small, you can fit in a couple of cassettes in total. But once it gets to a large enough size, it just becomes impractical to section the whole specimen. So what most labs do will provide representative sections. In neuropath, this is usually 1 per centimeter, but this what exactly is the definition of a representative section? will vary based on the clinical context. So some tumors where even a small component may be important for therapy, such as germ cell tumors, may require more extensive sampling, even if it cannot be done in toto. Whereas in other areas of pathology, 1 section per centimeter is usually sufficient to get an overall sense of the biology of a specific sample. So again, this really depends on the context of the specific specimen and what the implications are of a small subcomponent that may have an impact on therapy and outlook. So once you sample it, then you need to embed it in some hard medium to allow it to be cut. Usually, that's paraffin wax for light microscopy. The unfortunate part, though, is that, as we said, most of the fixative is water. And so, and paraffin is wax, right? So it's hydrophobic. So we need to now transition the tissue from this kind of aqueous state into a more hydrophobic state that will allow the wax to kind of penetrate the tissue and preserve it in this kind of firm medium. So to do that, we need to dehydrate with ethanol and then run it through an intermediary fluid called xylene, which is miscible with paraffin. This usually happens in kind of a tissue processor that can kind of robotically move the different fluids in the tissue. And so usually these are run overnight, and it's not something that humans need to do. So once they're found in xylene, they need to be oriented and put into the paraffin mold. So when we're sectioning them, we're sectioning the specific part of this slide that we are interested in. Now, most of this stuff is done in paraffin for routine samples, but it's important to note that you can also do this with kind of water-based medium, embedding medium called OCT, which is basically frozen into the hard substance that allows cutting. These obviously have a huge time advantage because you don't need to transition the tissue through these different substances. So they're really good for intraoperative examinations, but come with the downside that the morphology at the end, the quality is not as good, and obviously you cannot store them as easily as you could with wax. You would need to store them in kind of a refrigerator or a freezer, which is much more expensive than let's say a cupboard for paraffin embedded sections. So this is kind of what the machine looks like to embed samples. You have your tissue that's being sampled and put into these cassettes. These are labeled. The tissue is removed from each cassette and put into these kind of metal cassettes that are cold. The tissue's oriented, and then you have this dispenser here that's going to pour hot wax that's liquid into the mold. The cassette that has the label is put on top. Additional wax is put here to allow the allow it to stick to the tissue once it's frozen, and you can see this is the final product. So on one side you have the specimen label. You can also write it here, which is actually just engraved on in kind of a modern workflow with a computer, and then you have the frozen wax now from the mold that has the tissue oriented in the correct position that you'd want the tissue section. Okay, so here we then need to section and stain the tissue. So we put this mold here on what's called a microtome. In this particular case, the cassette was white, so just to kind of orient you. And then it's cut into very thin sections. You can see the outline of the wax, and then the tissue here is a different color. And you can kind of cut a number of different levels of the same tissue into these kind of strips. Because these are kind of very hydrophobic, they're not going to bind to a glass slide. So we tend to put them into this kind of water bath, which is kind of warm water. It helps kind of soften the wax and make them easily adherable to glass slides. So here the technician is picking up the sections with a glass slide and putting them here to dry. And that's basically the sectioning process. So again, it's cut on a microtome. We need to cut it very thin, 5 to 15 micrometers, put on a glass slide to allow the light to pass through the tissue. For EM, this is much thinner. It's 50 to 150 nanometers in thickness. So this is why we use different fixatives and different, we use paraffin for light microscopy. We use kind of a plastic resin for EM, just because it needs to be cut even thinner. So the material needs to be even thicker. And then we stain it, right? These sections, tissue does not have any inherent kind of contrast. It wasn't made to be looked at by humans. And so we need to stain it to be able to see individual cells. And so, again, when we stain it, we can make out histomorphology, the general structure, but we can also use histochemistry to look for the presence of specific components. To do that, we need to run the slides through these different solutions, as we'll see, and then stain it with the dyes. And you can see eosin is pink, and then this is hematoxylin, it's purple. Those are really the two components of the H&E stain. The key about the H&E stain, which differs with some of the histochemistry stains we'll see, is that it's a stain. It's like staining your shirt. The color of the stain will be the color that you find on your shirt or slide. So we'll see how these work in a second. So why are there so many of these buckets? And again, the problem is that the slide is inherently hydrophobic. We have the tissue is embedded in wax. So and the stains are hydrophilic. They're aqueous based stains. So we need to again transition the tissue that was originally aqueous, that we had to dehydrate to put into wax. Now we have to take it back into an aqueous solution. So the first step is called deparaffinization. We put it into xylene in this particular protocol, 2 times 10 minutes. This will remove the wax, and then we progressively put it into ethanol mixtures that contain more and more water. So this is 100% ethanol, whereas this is 96, 80, to try to allow water back into the tissue in a kind of gradual process. Then we put it into water. So that's the kind of final fully aqueous solutions. These concentrations can vary depending on the protocol, the number and the percentage of alcohol, but the idea is the same. We need to progressively move the tissue into a more aqueous state. And then once it's in an aqueous state, we can stain it with hematoxylin. And as you can see here, we have staining of certain components. Usually, as we'll see, it stains the nuclei, but it will stain most of the tissue with some sort of level of purpleness. There's different things you can do. There's a step called differentiation, where you will rinse the stained product with water. This will turn some of the purplish to a more bluish color, which is what we tend to see under the microscope as a final product. So that's called differentiation. And then you also want to no, that's called, I guess that's called that's not differentiation. Differentiation is washing it in water also removes some of the stain. So what you're doing is you're improving the selectivity of what's being stained. And when you're removing stain, usually there's progressive and regressive types of staining protocols. When you're removing stain through this kind of washing process, it's called a regressive stain, because you're regressing the amount of stain to improve the specificity. At the end, you get these blue nuclei, and you've washed out much of the staining of the cytoplasm, and then you use eosin to stain the cytoplasm pink. This usually stains proteins, which are abundant in the cytoplasm, and then you get this final product, which is kind of blue and pink or purple and pink, depending on how much of that bluization occurs. And now you have your stained tissue. Now, unfortunately, the cover slip that we need to cover and preserve the slide is xylene based. So we need to now dehydrate the slide back into a level of hydration that's more compatible with xylene and the mounting media. So we have to put it into, again, successively higher concentrations of ethanol, then into xylene, and then we can add the mounting media and the cover slip to have a finally preserved slide that can, stand the test of many decades sometimes, and allow you to store these in a kind of room temperature environment without losing any of the quality of the slide. So Hematoxylin Eosin, this is the most widely used stain in pathology. Again, the most versatile stain, because you can see not only cell types, but also cell patterns. Again, just some terminology, more just so you are familiar with these terms in case you have to troubleshoot. It's important to understand the components. So it's not the actual hematoxylin that's doing the heavy lifting. It's what is called hematin. This is the active colorant. This is what we see under the microscope, and it's usually combined with what's called a mordant or aluminum to help make it into a cationic structure that will allow it to react with the kind of basophilic structures of the tissue. So that cation is hemoglobin, and so that is what is staining the tissue, not hematoxin per se. So the stain, whatever you want to call it, is will complex with cell nuclei, DNA, and chromatin, specific amino acids on the chromatin proteins, and usually it's referred to basophilic, or those are called basophilic structures. Eosin is a kind of an acidic dye, and so it stains different types of components of the cell, usually proteins of the amino acids, and it'll stain them red, pink, and orange, depending on the type of collagenous tissue, the type of proteins, the amount that's present. So again, it's not just a binary pink, purple, pink, blue stain. There's a spectrum that you usually get between the two, and sometimes the spectrum is because some proteins or amino acids will actually stain with both, and then you're seeing the kind of mixture of the two together. Again, the staining is due to hemoglobin binding to the forming a kind of a dimetal complex with the arginine-rich basic nucleoproteins of histones. And so, tomato, it's good to know that if something is off, perhaps you have to look at the individual components to troubleshoot. Most of the cytoplasmic components are going to stain with eosin, as well as many of the kind of extracellular matrix components of the, that are rich in collagen and whatnot. Red blood cells stain red, as we can see here. This is a blood vessel, and we can see the red blood cells in there, and the capillaries staining bright red. And again, because of this spectrum, you get rich detail, right? It's not just a black or white. You actually see a very large spectrum that have incredible resolving powers of morphology. Sometimes you can see cilia. Sometimes you can see cell membranes. So you can see Golgi apparatuses or Nissl substance in the neurons. So very, very powerful. You may see other colors like brown and yellow. So it's important to note that there's some chemical components of tissue that are inherently, I guess, have chromogenic properties. Melanin, melanomas, other tumors have kind of melanin producing capabilities, or hemorrhage. Sometimes tumors will have hemorrhage in them, and then the kind of leftover residual heme groups will have their kind of own chromogenic properties. So beware that not all the stain is due to the actual staining. Does this state everything? No. Hydrophobic structures remain clear. Adipose tissue, fats, and in the CNS, the important one is myelin. And so we can do modifications to kind of bring some of those out. The kind of really nice one that we use in the central nervous system is the H&E plus LFB, Luxel Fast Blue, that allows us to stain and resolve myelin under the microscope with this blue color. So LFB blue, it's important to note that this doesn't work in the peripheral nervous system. It's unclear to my knowledge. Perhaps there's some different chemical properties of the myelin sheath that's formed by a Schwann cell and an oligodendrocyte. But for whatever reason, this stain is often said to work exclusively in the central nervous system. It's used to detect demyelination, and again, similar types of reactions. It's an acid-base reaction of the choline group in lipoproteins that replaces this that kind of interacts with these basic components or acidic components of the of this kind of amine group with the extra hydrogen. So this component of the Luxel Fast Blue dye will bind to choline and lipoproteins. These are exceptionally high when the oligodendrocytes and are wrapping around the axons to form their myelin sheaths. So these areas are rich in phosphatidylcholines, so they're going to stain particularly rich for this blue color. And so here we're seeing different tracks in the corpus striatum, probably where we see these rich bundle fibers, and these would be kind of attenuated or lost in diseases like multiple sclerosis, where you have demyelination. So if it's so pretty and so useful, why don't we do this for every section? Well, there's two reasons. Sometimes the blueness can be distracting if you're looking for mitoses or other components in tumors or whatnot, where the blue does not provide any diagnostic value, and this stain occurs overnight, so it just takes too long. So we don't want to do that, particularly on every single section. Just briefly, let's look at some histochemical stains. A lot of these are silver stains. One of them is the Belchowski stain, and really the key thing here is that instead of having a dye that is binding to specific acidic and basic components of tissue, you now have silver ions or cations that are binding to proteins. And the important difference here is that these are should not be thought of as I guess dyes, because the substance is clear, unlike the eosin and hematoxylin that we saw was pink and purple. Silver stains are actually clear, so they do require kind of a reducing step to convert that that metal into something that can be visualized under the microscope. So there is an additional step that's important to know about. And again, the reason why this works is that the silver stain preferentially stains proteins with basic amino acids, lysines and histidines. And so this, for whatever empirical purposes, works very well for protein aggregates. This will work well for plaques and Alzheimer's disease, as we'll see here. So it's very useful for specific use cases, such as quantifying the disease burden in Alzheimer's disease. Again, just a kind of simple chemical reaction, just so we get an idea of how this works. You have this protein structure here. It will bind with the anion of the silver salt, and then it will be keen to and accept the positive charge of the silver metal. It'll form this bond, and then this will relieve the kind of chemical charge imbalance of the here you produce your acid, and then you have your silver metal attached to specific proteins, which then you can reduce with sodium hydroxide and get that brilliant staining that you see here. So this is, again, meant to complement really the H&E stain. Very useful in certain situations, but you need the H&E to guide you about what those use cases are. So you're not doing these silver stains, which you can imagine are expensive. They have silver in them on every single case. Now, again, just to briefly end, this is not going to be a thorough discussion, as we have other lectures on NeuroPsyQ that cover computational pathology basics, and we'll make many more. But really, the key thing to understand is, why am I sitting through a lecture on an H&E stain in 2022? And the reason for that is that there's been aside from the powerful aspect of it, even in the hands of an experienced pathologist, there's been an emerging and explosive interest in the H&E stain, because it's been amenable to modern innovations in computer vision, such as deep learning. And what these innovations attempt to do is to remove the subjectivity, or that's the goal, the end goal. We're still very much in their infancy, is to remove the subjectivity of the H&E exam, or anything that requires subjectivity in medicine, where my interpretation may differ from my colleague or even myself based on recently read material, the degree of fatigue I'm experiencing. So we want to remove those factors. We want to remove the patient-dependent factors from medical decision-making, and that's really what AI is all about, is providing some objectivity and automation to the system. So you can see here, you can take tissue, and these are hold slide images, so probably 2 by 2 centimeter sections, gigabytes of pixels, right? I would have to look at this very carefully to kind of quantify and note where and what types of tissues there are. But even very basic computer vision workflows can kind of give you a rough idea of how much necrosis is there, where the normal and abnormal tissue is. Here in black, we're highlighting the kind of infiltrative glioma. The gray is kind of more gray matter, and blood is red, and pink. This magenta color is necrosis. This is a glioma. This is a disease called meningoangiomatosis, where you have a meningioma infiltrating the cortex, and you can see it here. And then you see gray and white matter being classified appropriately. And then you get a quantified value of what exactly you're looking at. So very, very powerful for classification. In some unique cases, these will work as good as pathologists. Again, this still needs to be expanded to infections, different types of tumors, different subtypes, but it does seem to have the potential to automate some of these subjective tasks. The other big area is in immunohistochemistry, or image segmentation. This also works on H&E, but one of the problems that we have in pathology is the need to estimate things like proliferation indices in tumors, and quantify PD-L1 expression for immunotherapy. PR statuses in breast cancer. So, this takes a lot of time to do this properly. You can eyeball it. Sometimes you may be off, your estimate may be off, or counting them is a very laborious process, right? So image segmentation tools will not only identify, but can kind of bin and count the positive and negative events in a specific slide and allow you to do this automatically. So this is really cool because now we can use these as biomarkers to see if the exact percentage is kind of a prognostic marker of outcome or disease response. Another cool area, which we talked about in a previous lecture, is image registration or image alignment. As pathologists, we often are comparing the H&E and the immunohistochemistry slide side by side to try to see how much positivity we have, not in the entire slide, but in areas where there's actually viable tumor or tissue of interest. And this is laborious because you got to go to the H&E slide. You got to align it kind of in your brain with the immunohistochemistry slide of interest, and then try to estimate what proportion of a slide is positive. What computational pathology will eventually allow you to do in a kind of routine way is to align these slides together so you can examine these together in a kind of more harmonious manner. One last application that I'll mention, which we will talk about more in detail in a future lecture, is this concept of tumor mapping, and being able to find subclones within a tumor that may have different biology. So here we have these 3 sections. We can analyze this with a computer. These are largely brain tissue infiltrated by tumor cells, whereas this is more kind of homogeneous tumor that that we would consider a glioma. So you see, when we use computational tools, we can actually map different morphologies that the computer is perceiving in this section, and then we can assume that if it's subdividing areas that we thought were tumor into subcomponents, there must be some biological basis for this, because as Virchow said, chemical changes often manifest as phenotypic ones. And so if there's phenotypic changes that we can now kind of precisely localize using kind of automated computer programs, we can now potentially examine these areas differently than other areas of the tumors that have different morphologies. And as you can see here, when we look at the kind of proliferation indices in these subcomponents, we see that these orange subclones have a much higher proliferation index than other areas of the tumor. And so perhaps there's some different biology that we need to iron out in these subclones that we may miss if we only examine the kind of cyan and blue area of the tumor. So this is not meant to be exhaustive or detailed, just to give you an idea of some of the exciting things you can do with H&E stains, and we'll talk about them in more detail in future episodes. So with that, I want to say thank you for listening. Thanks for supporting the channel. Stay tuned for a lot more content about pathology, computer vision, and AI.